Yoël Forterre, welcome. So this seems surprising that this wasn't figured out long before now. Were you surprised when you first heard that the Venus flytrap snap was a bit of a mystery?
Absolutely. Thank you very much first for inviting us. And yes, I discovered the plant like 20 years ago when I was postdoc in a lab in Cambridge, UK. And a botanist friend gave us these plants, and this is the first time I could imagine and see with my naked eyes that plants could snapshot so fast. And since then, I've been completely obsessed by the idea of what is the engine of this motion. I mean, because you know, plants, they do not have muscles and nerves like you and me. I mean, they have to find another way, and they are very efficient in this. So this is kind of a fascinating system for us, even as a physicist. I mean.
Yeah, and 20 years later, after this obsession started, it seems like you've sorted it out. So explain for us the setup of your study.
Well, first, the idea was to dissect the trap of the plants in order to really see what's going on inside. I mean, what is the dynamics of that internal muscles, green muscles that the plant is using. And by doing this dissection, we could unveil the dynamics of the closure, the time scale, and then we try to see what is the mechanisms for that. Because you know, plants, they rely on water. They bring water from the soils to the leaves, and usually plants, they are using water in order to swell their cells, to change the volume of their tissue. So it was the first idea. But then it turns out that when we measure the time it takes for water to move in that trap, it was much too long to explain it. So we turned to another hypothesis, brings about more than 20, 30 years ago by German scientists who proposed that perhaps it could be that the cell wall, the materials of the plant itself, that could be actively tuned by the plant. And it turns out that it is true.
Yeah, and so how certain are you that the source of this snap is the relaxing cell wall?
I mean, this is a kind of a measurements we've made. I mean, we poke with a machine on the cell, a little bit like if you poke on a balloon. And when you do this, as soon as you trigger the trap, what you see is that the balloon, let's say, becomes softer. So that's a kind of a direct measurement. But it is true that this balloon could become softer because either the pressure inside the balloon decreases, or either the balloon itself, the rubber materials, becomes more flexible. And that was kind of a trick in order to disentangle this. And we had a kind of indirect clue that this is indeed the wall that becomes more flexible. But for sure, more work is required to find, importantly, the molecular reason for that rapid softening, which is the fastest so far that we have observed in plants.
Yeah. Now, as with all scientific studies, there are other scientists with opinions, and I'm sure as a scientist you welcome that. One here
sure.
in Australia argues you didn't test another mechanism for water transport, that the water in a line of cells could move simultaneously along for faster transport. What do you make of that observation?
Yeah, so actually we tested this. I mean, we directly measured the time scale it took for tissue to swell, and this incorporate both kind of transport mechanisms that you are talking about. So, I mean, we are confident that this time scale is much too long to explain the closure. But as you said, I mean, only future works will really confirm this kind of stuff. I mean, in science, you really need not only one study, but you need several studies to really consolidate a result. But we test this idea of water, and definitely it took like minutes for water to swell the tissue, whatever the pathway, water can be simultaneously or not. I mean, it has to move from one place to the other in order to create this closure motion. So, yeah, that's what I can answer. But I understand. I mean, I understand the discussion.
Indeed. And so if your theory of relaxing cell wall holds up, what do you hope it could lead to?
So, well, there are two directions maybe. For the biology, so as you said, I mean, I'm a physicist. So, I mean, this is not my expertise like molecular biology, genetics, but recently biologists start to produce transgenic Venus flytraps. So they are going to be able to manipulate the molecule inside the plants, the genes, in order to test some of the fundamental biological mechanisms behind this. So that's one very important thing that we have to do ahead. The second thing is that more from an engineering point of view, I mean, the Venus flytraps combine a very sophisticated traps that release very quickly stored elastic energy by actively tuning its materials. And this is really new for living organisms, and I think this could lead to good ideas to generate materials that change actively and fast their shape, for instance. Yeah.
Yeah. Okay. Yoël, it's fascinating hearing you talking about this. Congratulations on pursuing your obsession, and thanks so much for talking to us.
Thank you very much, Joe.